Okay, hello everyone. Welcome to our webinar. My name is Liz Meyering. I think we'll get started. People are still coming in. I'm going to give a bit of an introduction before we go to the talks. So I am President-elect of the Protein Society, and I'm really glad to be introducing this really exciting webinar. This is our final webinar before we break for the summer. And our next series of webinars will start in the fall. So I want to invite everyone to consider participating in the webinars. You can suggest topics or organize one yourself if there's an area that you're particularly interested in. We've had a wide range of different kinds of topics for our webinars. They're all available on YouTube. So you can either suggest a topic or post one yourself and give a talk on an exciting area that you're interested in. So these webinars are actually really easy to do. We have wonderful staff at the Protein Society who take care of all the organization, Raluca and Shannon. And so really at your part of it is just to think about the science, and you can think of 3 or 4 people typically. That's how we can organize these things, but that also is flexible. So you can contact any of us. You can contact me, Liz Meyering, or the staff at the Protein Society using by email, and you can get that at the webpage for the Protein Society. So this webinar is on CryoEM of molecular machines, current capabilities. Let me just get my laser pointer. There we go. Okay. This webinar was organized by John Rubinstein, who's at the Hospital for Sick Children in Toronto. And our speakers are Kelly Nguyen from the MRC Lab of Molecular Biology in England, in Cambridge, and Yulia Mahamid, who's at the European Molecular Biology Laboratory in Germany, so in UBL. So what I'm going to do is give a few announcements before we proceed with the science. So I just wanted to remind everyone that the Protein Society has various activities in addition to the webinars. So I want to invite you to join us at the 37th Annual Symposium, which this year is in Boston and taking place from July 13th to 16th. Today is actually the last day for discounted rates and abstracts, so you can still sign up. And here is the website where you can get lots of additional information. So I look forward to seeing many of you there. I also wanted to highlight that the Society has a journal, Protein Science. John Nguyen is the chief editor, and we want to encourage you also to consider submitting some of your excellent work to this journal. It's got a long-running journal that highlights a wide range of protein science. Okay, so turning to the topic of our webinar, CryoEM. John Rubinstein, unfortunately, actually can't be with us today. He had an unexpected personal thing come up. So what I'm going to do is give a little bit of information about his research, sort of an overview, and then turn it over to Kelly and Yulia. So I have some slides on this. This is from YouTube, actually, that I nicked because John wasn't able to send me this. But this is a current presentation he gave, and this webinar and the other webinars from the Protein Society are also on YouTube. So John obtained his PhD from the University of Guelph. Then he did his PhD in Cambridge, where he worked at the LMB with Sir John Walker and Richard Henderson, starting to work on ATP synthase by CryoEM. He was also a postdoc there for a time, and then he did another postdoc at Banting and Best Institute for Medical Research in Canada before starting up his own group at the University of Toronto. So a focus of his work is macromolecular machines. I feel like my pointer keeps disappearing, doesn't it? Every time I do something here. Okay. So we're working on molecular machines, large machines at energized membranes. So ATP synthase, and ATPase proton pumps. So here's an image to give you a bit of a context for where CryoEM has been and gotten to. This is from John's PhD work, culmination of his PhD work. And it's the 32 angstrom structure of the mitochondrial ATP synthase, which was a very hard project. And his work since then, you'll see in the next slides, has really gone to amazing increased resolution. So in his group currently, they have a pipeline, which kind of highlights how once you have a good way of making the samples, you can actually then get to getting a structure in 2 to 3 weeks. This is an amazing advance, if you think, relative to his PhD paper in 2003. So here are a variety of type 5 ATPases from different organisms that John's team has worked on. Very important fundamentally and as increasingly viable targets for disease and for treatment of disease. Here is the ATPase synthase structure. So we had the ATPase, now we're going back to the ATP synthase. And so relative to that blob that I showed you at 32 angstroms, these are the kinds of details that are now known about this machine. And in terms of understanding how this structure relates to its function, we really have amazing high resolution molecular details. So here's a model of how the synthase proton shuttle would work from long ago, 1997. And then now it's actually known what exact residues are involved in shuttling the proton and how these conformational changes occur and how the whole molecular mechanism works. We're still getting insights into what subunits are involved at different stages, how you can alter or target different sites here, also for developing drugs. So really phenomenal high resolution mechanistic information. So that is kind of the higher resolution or more smaller scale CryoEM aspects that John is working on. Turning to our speakers today, Kelly Nguyen did her PhD at MRC LMB with Kiyoshi Nagai, working on the spliceosome. Then she went and did a PDF at UC Berkeley, working with Kathleen Collins and Eva Nogales, working on telomerase, and then moved back to the LMB just pre-pandemic, where her team is working on the structure and function of telomerase and has really continued to elucidate critical roles for this machine in molecular aging and disease. And that's what she'll be telling us about today. Going even bigger, Yulia Mahamid got her PhD in chemistry at the Weizmann Institute of Science, working with Leah Adadi and Steve Weiner on biomineralization in bone using CryoEM. So a very different kind of a system in aggregates. And that's kind of one of the amazing things about CryoEM is all these different systems that we can now look at. She then did postdoctoral research at the Max Planck Institute in Germany, working with Tony Hyman on biomolecular condensates. And she has been a group leader now at the EMBL since 2017. She's an ERC investigator, and her team is working on biomolecular condensates and state-of-the-art cellular cryo-electron tomography. So the previous work was a single particle analysis, but you can use tomography to look at the whole cell and advance our understanding of functional organization in the cytoplasm. So I'm really excited to hear about these advances. I want to thank our speakers. Thank you, Kelly and Yulia. I also want to thank John for organizing and our team at the Protein Society, Shannon and Luca, for organizing. The way this is going to work is each of our speakers will give roughly a 30-minute presentation. Then we'll have time for discussion. So you can type your questions in the Q&A box. So look for that and post it. And then I will read the questions out and our speakers will answer the questions. So again, thank you for all your participation, and I'll turn it over to our speakers.
Thank you very much, Liz, for the kind introduction. I'm gonna try to share my screen. Can you all see my screen? Okay, perfect. Okay, and I will turn this place pointer. Okay, perfect. Cool. First of all, I would like to thank the Processing Society for giving the CryoEM community a chance to have our webinar as part of the very wonderful series. And also John for organizing despite the fact that he can't be here today. So what I'm going to tell you today is how we're using this technique called EM to visualize this molecule here that Liz has mentioned called telomerase. And so the main goal of what I'm going to try to get across for my talk today is two main points. So one is, why do we study telomerase, why it's important, and why CryoEM was very instrumental in our understanding of this molecule that, it's called telomerase, that you already heard. So to begin with, I'm going to give you an overview of why we study telomerase first. And my lab at OMB focuses on this region of the genome called telomeres. And as you can see here, this is a metaphase spread when you can see these condensed chromosomes labeled with that being blue. And the dots that are located at the end of chromosomes, these are called telomeres. And now if we just look at it a bit more closely to see how telomeres are structured, it's quite different from the rest of the genome. So in most eukaryotes, telomere generally consists of G-rich repeats. And in human, this is 6 nucleotides, GGTTAG. And this can range between 2 kilobases to 100 kilobases in a mammalian cell. And it also had this 3' overhang, as shown here on the G strand. And along this telomeric DNA, what we know is that in mammalian cells, there's a complex called the sheltering complex, which can specifically bind this telomeric DNA, both at the double-stranded part, as well as the single-stranded region as shown here. And together, what we kind of think about telomeres is that together with the DNA, it's formed like a protective caps that protect our genetic information from undergoing degradation and also inappropriate break repairs. But now, if you look at how the chromosome ends are structured, as I mentioned, it has linear ends, and this is actually problematic for the ribosome to replicate completely. So this brings us to a problem that we know as the end replication problem. So every time the cell divides, we lose a bit of our telomeres, and this leads to a gradual shortening of telomeres over time. In a young cell, there's plenty of telomeres, and it gets shorter until it reaches about 2 kilobases or so. And this is known as the famous Hayflick limit. And then when this happens, this will lead to a range of phenomenon, including genomic instability, growth arrest, and eventually cell death. The cell can no longer divide, and they eventually will undergo apoptosis. And this happens for pretty much all the somatic cells in multicellular eukaryotes. And you can think about it as most of what is in our body. And the question is, how do cells overcome this problem? And the way they overcome this problem is by this enzyme that we call telomerase. And telomerase is actually only expressed in a very small subset of cells in the human body, and that includes cancers, stem cells, and germline cells. And by turning on telomerase, they can compensate for the telomere shortening effect that I told you about. So you can already guess what telomerase does in the cell. It adds telomere back, and therefore these cells can maintain a stable telomere length over a very long period of time. And this is one of the reasons why you can culture some of the cells like HeLa cells or HEC cells in culture, because these have telomerase turn on. And this is also why it is often regarded as the immortality enzyme. So with this knowledge, what has been known for many years since the discovery is that it's a very potent therapeutic target. And you can think about it in multiple ways. One is that, as I mentioned, is activated in a very small subset of cells, including cancer. So inhibition can be used to target cancer cells. On the other hand, if you can activate the normal cell, which normally doesn't have telomerase, then one can think about a way to prolong the age of the normal cells. And also what we know is that there are a number of disease mutations that compromise telomerase function in the cell. This actually leads to a range of premature aging disease pathologies. So that's kind of in multiple ways we can look at how we can use this as a drug target. But as we speak, there's no inhibition or activation of telomerase for disease therapy available in the market. And one of the challenges that has been for drug design targeting telomerase is the lack of information on its structure. And this is where our work come into play. And so as a nutshell, as a structural biologist, we really want to understand how it works at a molecular level and what the way it does this DNA addition to the telomeres is like via a subunit called telomerase reverse transcriptase. But unlike retroviral RT, telomerase is special in a way that it also contains an RNA component, and this is called HTR in human. And this RNA had this region, a very small region that we call the template region, and half of it can specifically find the end of the DNA by just very simple base pairing, as you can see here. And then the other half of the template is then used by the reverse transcriptase to synthesize the exact GTTTAG repeats. And furthermore, this is also special about this enzyme is that it can do this multiple times every time it binds to a DNA target. And this processivity is very unique to telomerase, and that is called repeat addition processivity, or RAP. And the cell does need this processivity in order to survive long term. So this is a very important property for telomerase. And in the lab, we usually capture this repeat addition processivity using an in vitro assay, which you can see here on the right-hand side. When you give telomerase a DNA substrate and DNTPs, and one of them is p32 labeled, and when you let it extend the DNA, and what you see when you extend this DNA is that you see 6 nucleotide repeats being added to the DNA. So with that in mind, you now know that telomerase needs TERT and HTR to do its job, but the picture in the cell is a lot more complex than just TERT and HTR. And to start with, I'm going to kind of give you a quick tour of what the RNA scaffold look like. So this is a secondary structure prediction of telomerase RNA. And as a structure biologist, we like to do things in domains. And the same thing, you can divide this RNA into 3 domains. So the first domain is what we call the template pseudoknot domain here that has a template region for DNA synthesis and a region that we call conserved regions 4 and 5. And the RT is known to bind these 2 domains of the RNA. And at the 3' end of the RNA, you can see here in purple, there's a 3rd domain that is called the HACA domain. And this was proposed to be bound to various proteins to call HACA proteins. For the purpose of today's talk, you don't need to know too much about what these HACA proteins are and what they do in the context of telomerase or the cell, but just kind of keep in mind that they are there. But this kind of composition of telomerase is, for many years, for 2 decades, has been kind of controversial in the field. So the question of what's in human telomerase has been a matter of debate for many years, and there's many different proposals of what's actually in the enzyme. And so in order to address this, I kind of was very fortunate to be offered a postdoc position with Kathy Collins, who is a telomerase expert, and Evan Nogales, who is a CryoEM expert, to use CryoEM to address this problem by visualizing telomerase itself. So this is where the project was started. So telomerase, as I mentioned briefly, is absent in most of the cells, and it's actually one of the very rarest enzymes in the cell. The first challenge to actually studying this structurally is actually reconstitute and purify to the quantity that one can think of structural studies. And that's the first kind of major step forward that I did when I was a postdoc in Kathy and Eva's lab, is to devise this reconstitution. The way we do it is quite in a simple term, is that we use a semi-endogenous system to transfect in HEC293T cells, just limiting factors. In this case, is the reverse transcriptase as well as the RNA, and everything else come endogenously. And then because it's very, very, rare and difficult to assemble, so by growing quite a lot of cells, if we prep is hundreds of plates of transfected cells, we're able to reach the quantity that one can start thinking about doing structure biology on, which is a few microgram. And again, using silver stain, we can actually see the complex with all the subunits that has been previously suggested to be part of telomerase. And again, we have this in vitro assay to test whether our reconstituted complex is active. So you can see here on the right, our purified telomerase is highly active by extending the DNA very well. So now, guided with this sample, the next question we ask is how do you then visualize this enzyme? And as many of you know, that visualization of biological specimen is a very powerful way to understand how they work. And here is a spatial scale of different objects on the top here, ranging from atoms all the way to insects, where you can see with your human eyes. And the bottom is the different techniques that one used to visualize these different objects. And as I said, human eyes are very good at seeing big things, but when you start looking in micron scale, you start needing more specialized technique. And the range of the molecules we're studying are macromolecules that include proteins, DNA, RNA, and that's in this range here. And that's when there are three main techniques that are commonly used to do this. And they are X-ray crystallography, NMR, and electron microscopy. And the question is, which is the most suitable for human telomerase? So there's a number of pros and cons one had to consider for this case. And what we then decided, obviously, now talking about EM, so it was the methods of choice. I'm going to tell you a little bit why it is really, the main methods that can be used for studying this enzyme. And one of the main advantage of this CryoEM method is it can work with a very small quantity, and this I mean microgram scale of sample, and that's the range we are in for telomerase, what we can make. Whereas for NMR and X-ray crystallography, you need to go milligrams in terms of sample quantities, and that's really out of the reach of what we can achieve. And second is that for CryoEM, the bigger the particle, the better the signal. So it's actually the opposite for NMR and X-ray crystallography. It's a lot harder to study large complexes by NMR, and also difficult to crystallize them. And last but not least, and that has become very important for our work, is that we can deal with compositional and conformational heterogeneity using a lot of computational image analysis. In this case, our sample is very heterogeneous in terms of conformation. So having all of this kind of knowledge, why we chose this, I'm going to quickly give you an overview of single particle cryo-EM, what this technique involves. So first, like all the structure technique, you need to start with sample preparation, and we need a good complex that kind of purified to as best as we can. And then what we do in EM is we apply the sample on a small grid. It's very tiny, it's like 3 millimeter across, and about 3 to 4 microliter generally, using the conventional method we have been using. And then what we do is that we plot most of the sample away, so leaving a very thin layer of your sample on this grid. And then we plunge it very quickly into cryogen, in this case, the liquid ethane. And the trick is that if you freeze the sample so quickly that the ice crystal doesn't have time to form. So we have eventually on this grid is that they have this class C state of ice, which we call vitrified ice, where your particles are embedded into this ice layer. It's almost like your window with the particle embedded within your glass window. And having done that, we then put it on the microscope and then shine electron beam on the microscope and then collect data. And that brings us to the second part of cryo-EM. A single part of EM is image processing, where we collect a lot of these micrographs, which we call electron micrographs. When you have the particles on these micrographs, you can see here, but these are very low signal to noise. So what we have to do is collect a lot of them and pick these particles on the micrographs and average. So invert the contrast and average many of these in order to improve our signal to noise. And this is what we call 2D classification. And based on these class averages, you can see here, we can see the particles in different views. And then we perform another procedure called 3D classification, when we can sort these particles in different 3D states. This can be compositional or conformational. And then we choose the subset of interest to reconstruct a structure, three-dimensional structure from these two-dimensional projections of the particle. By doing this, we can then get our final structure. So having done all of this, so EM has been around for many years, so it's not a new technique. But in around 10 years ago, 2013, the field underwent what we call the resolution revolution. And this is thanks to developments in direct electron detectors as well as statistical image processing. So together, the field really make a big jump in the resolution that we can achieve. And back then, there were structures of various samples about 10 or 9 years ago. And for some examples here that I took out of a review written by Werner Kuhn-Braun in Science, that we can now achieve, 3 to 4 angstrom resolution for various samples. It's extremely powerful. But at the same time, this is what we can achieve with these ribosome, membrane proteins or hydrogenase. The same year, when this happened, we also have a structure of human telomerase, but this is still in the negative plane, kind of domain. And the structures you can see here give us a first glimpse into this how things are organized. But as you can see, compared to these, we are at 30 angstrom resolution. So there's a big gap between, what we could do in the EM field and what we have for the telomerase field. And this is where I'm going to tell you a little bit how we get there and where we are now. So then applying the methods that I mentioned, using these direct electron detectors as well as the image processing techniques, we apply for telomerase bound to DNA substrate. And here are some of the 2D class averages. And as I mentioned briefly before, it's actually very conformational heterogeneous. So composition is fine because it's very defined, but the structure has the elongated shape and is really kind of flexible. So the way we overcame this is just by collecting a lot of data, in this case about 1 million particles, and then sort out a lot of this by image processing to achieve at about 8 angstrom resolution from about 3 to 4 angstrom of percent of the data. So the structures had this elongated shape, and we call this top lobe and bottom lobe, and we treat them as independent entities doing image processing. But together, this allow us to achieve kind of first big step towards understanding how things are organized. And you see that this is still far from 3 to 4 angstrom resolution, but this still tells us a lot about the composition and also the architecture of the enzyme, which I will go over, give you an overview of what we learned from this work on this slide. So I'm going to start with something that everyone has seen on the previous slide, which was proposed to be in human telomerase. And then I'm going to try to turn into what we see in the three-dimensional structure as shown here. And perhaps for me, one of the most remarkable things when I see the structures is that the whole structures is scaffolded by telomerase RNA, which are shown in different colors here. And the protein kind of stick together via the RNA, a lot of them. And now let's turn our focus into the protein of the complex. And here on the right is our CryoEM density. And here I'm coloring the RNA as one color, is in blue here, and it's shown in this cartoon on the right. I'm going to walk you through how the proteins are organized on the RNA. So you have the RT, the reverse transcriptase, bound to the bottom, and the DNA substrate also bound to the RT. And then on the top of the structure is the HACA proteins. Here we have 9 of them as shown here. And again, you don't need to worry about the names, just know that they are there. And so based on these assignments, we can see the structures had 1 RNA and 10 proteins. So it's really nice to see the composition of the enzyme by just looking at it. And also based on the structure, what we can also see is these proteins are kind of segregated into 2. Each of the part has a very different functionalities. The top lobe we call the HACA lobe, and is important for biogenesis and localization. And the bottom lobe is what we name the catalytic core, because it's important for DNA synthesis, because it has the reverse transcriptase, and also later I'll briefly touch on the recruitment to telomeres. So yeah, that's kind of what we learned from this structure. But again, as I said, this is very far from 3 to 4 angstrom. So the next question we ask is, how do we improve the resolution? And in a short, kind of the short answer for doing this is a number of things. So we then employ the newest technology in detector, as well as collect many times more data, this time physically 15 times more particles, and spend more time doing computational work to sort all heterogeneity out. So together, using these 3 things, that allow us to make the big jump, where we can obtain the atomic structure of this enzyme, and which I'm going to show you in this brief movie. And this is the work done by 3 very talented people in my lab, shown here in collaboration with Richard Das and Kathy Collins, and all in the middle of 2020, when we were all at home, actually. So I'm going to start with, again, what you have seen before, with the 8 angstrom structure. And then again, we have this divided into 2 parts, HACA lobe and the catalytic core. And then going to morph it into the new structure now, which are below the 4 angstrom. And hope you can appreciate the details that we have been able to see by this resolution improvement. And at the bottom of the structure is what we call the catalytic core. Again, we have the TERT bound to the RNA and also a pair of histones. In the center of the active site, we have a DNA substrate. So because we can model the entire provide atomic model for this, we can actually see how the DNA interact with telomerase in the active site. And also by solving the structure, we also discovered new subunits, which are in this case histone H2A and H2B. I'm going to touch a little bit on that after the movie. So you can see there's a pair of histones. And we also have high resolution structure of the other part of the enzyme, the HACA RMP. And this is actually, I didn't mention, but this is one of the very first kind of eukaryotic HACA RMP that's part of a big family. So we just have 2, we get 2 targets in one shot. And we also together, we have a structure that has low resolution, but the 2 lobes are together. And in collaboration with Richard Das, we can build the RNA linker between the 2 lobes, and that allow us to have a complete atomic structure of this enzyme, in this case with 12 protein subunits and telomerase RNA. So there's a lot of details one can gain from this, but what I would like to highlight is one of the discoveries we made by the structure, which was not predicted by any previous genetic or biochemical work, and that is the identification of the histone subunits here. So previously in the 8 angstrom map, we have this density that was in model, and it's always there, and we couldn't have the resolution to tell what it is. But using this high resolution map, we can then, ask a very general question, very unbiased. Given this density, what in the PDB will fit this density? Because we tried out the literature and we couldn't find what has been previously to interact with telomerase. We couldn't fit anything else in here. So then we using this unbiased map and an X-ray crystallography technique called molecular replacement, and that allow us to identify histone H2B in this density. So here is that unmodeled density, and here's histone H2B in this PDB in pink. What you can also see is that it only fit half of the density. So if you take H2A from the same PDB, put it into the map, and then it fits perfectly. So then that kind of gives us then an updated view of what is the telomerase catalytic core, which not only contain the reverse transcriptase and the RNA, but also a pair of histones. So we were really bothered by this because we all at home couldn't do the experiments. We asked ourselves, what are they doing in telomerase? And it turns out that many DNA binding proteins also bind RNA. And this is the answer in this case, what the histones are doing in our structure. So the histones are binding a highly conserved motif in telomerase RNA, and it's called conserved regions 4 and 5, because again, you see it's very conserved. And this is this region here. And this region forms what we call a three-way junction, as shown here, and the histone binds across two standard three-way junction. And you might already remember from previous slide, the reverse transcriptase bind the same region and actually bind on the other side of the three-way junction. And what is also remarkable that we didn't expect is that together they seem to bind and shape the stem loop here that has been shown by many previous genetic and biochemical studies to be essential for telomerase catalytic activity, because if you mutate this or disrupt the structure, telomerase cannot function. This seems to be very important for folding this domain of the RNA. And not surprisingly, the way it used to bind the RNA is, as you can see here in this surface charge, it used a very positive charge surface to bind RNA. And also remarkably, if one put in our structure side by side with the nucleosome structure, when the histone dimer oriented the same way, it binds RNA in a very similar way that it binds DNA. So again, this is all unexpected, and we're still kind of trying to nail down the actual roles in telomerase and whether they have any general role in RNA biology, given that the binding is quite simple. And still, hopefully, in the near future, we'll be able to tell you a little bit more about that. And taking this a step further, having studied telomerase structure on its own, we also want to understand how it's regulated in cells. So using the same methodology we developed, we then start to study telomerase regulation in cell. And as I did not mention, actually, so is that most of the time in the cell, telomerase is not located at telomeres. And in mammalian cell, telomeres are occupied by a different complex called the Shelterin complex, which I also briefly mentioned, but this composition is shown here, which you don't need to worry too much about. And telomerase is often located in a different location in the nucleus called the Cajal body. So only a very specific stage of the cell cycle that at the end of the S phase, the telomerase get recruited to the telomeres via the Shelterin complex itself. And this is what we call telomerase recruitment. And this is very essential for this activity. So without successful recruitment, there will be no telomere lengthening by telomerase. So it's a very important step of regulation. So out of all of these subunits of Shelterin, work by many labs have shown that one of the subunits called TPP1 that directly recruit telomerase by direct interactions with the reverse transcriptase itself. And beside this, there's also another factor called POD1, which is essentially a DNA binding, single-stranded DNA binding protein. And together, they have been shown by many previous work in the field to stimulate telomerase positivity. And this can be very nicely captured in vitro because we turn into our in vitro assay again. So just to remind you, when you have DNA and telomerase, you get this ladder of repeat synthesis. And then if you add, the more repeat added, the more, darker DNA would you see here. And if we add this Shelterin to our telomerase at higher concentration, I hope you can appreciate that telomerase can make much longer repeats in the presence of Shelterin. And at the time, the structure basis the recruitment, and so the stimulation was not known. So we thought we can use the methodology we already developed to address this problem. So having done this is the work done by a very talented pair of postdoc and student, George and Zala in the lab. So we solved two structures. So we then solved what we call the human telomerase recruitment complex. And in this structure here, these two factors binds at the catalytic core, as shown here in this cartoon. So we saw the structure of the catalytic core first with TPP1 and DNA. And then here is the structure. You have telomerase on the top that you have seen before, and TP1 bind at the bottom. And then the second structure, we have pod TP1 and pod 1 bound to telomerase in the DNA. And here is the structure when you can see, in addition to PP1, you also have pod 1 binding in front of telomerase catalytic core. So what I would show you is just a summary of this work, show you the structures and kind of briefly how we think that these factors recruit telomerase and simulate this processivity in this movie here. So I'm gonna start with the TPP1 telomerase structure when you have T1 at the bottom and the subunit of telomerase on the top. And then using this, now we can build a full atomic model for this, and we can actually zoom in to see how TP1 interact with telomerase at a molecular level and really explain some of the observations that identify these interaction regions between TP1 and telomerase, as well as revealing new insights. And the second structure is with T1 pod 1, and pod 1 is literally just bind in front of the telomerase active site. You can see here on this region, and TP1 stay in the same place. And here you can resolve a DNA because pod 1 binds single-stranded DNA. And what we found based on the structure is together, the binding of TP1 and pod 1 to telomerase stabilizes the DNA substrate on telomerase. You can see here in the previous structure, we see only 6 nucleotides up to here. In this structure, we see the entire path of DNA coming out of the active site. So what we propose that these two factors kind of stimulate telomerase processivity because it allows telomerase to stay on the DNA more stably rather than falling off. And that's how they both recruit as well as increase the processivity of telomerase on the DNA. So also for this work, we collected a lot of data that allow us to actually resolve the structure of the other lobe of the enzyme, which didn't undergo huge conformational changes. But this, we resolve it to now 2.7 angstrom resolution, and this is our density. And this lobe has a lot of functioning other pathways in the cell. So this is actually quite important for another field in addition to telomerase. But this is really the first human structure that went below 3 angstrom resolution, and then it's currently the high resolution structure of any telomerase to date. So this is kind of almost a dream come true for us. When we started this work, we never think that we could get this low. So to summarize, I'm going to kind of give you an overview of how CryoEM has really enabled us to visualize this enzyme in ways more details than we had just about 10 years ago. So as I mentioned before, 10 years ago, we see the first glimpse of telomerase at 30 angstrom resolution. And then in 2018, we make a big jump to 8 angstrom where we can see the composition and the architecture of the enzyme. And in 2021, we are able to now bring it down to 4 angstrom when one can actually see atomic details of the enzyme, and then also discover new subunits just based on the structure analysis alone. And last is now this year, we managed to bring it down to 2.7 angstrom resolution, not just only telomerase, but also the regulatory factors, in this case, the recruitment factor you can see here. And I was also want to highlight that the work done by other labs, including Mingley and Julie Feigen, and they reached very similar conclusions to what I just described to you. And with that, I would like to acknowledge many people who have been, making a difference, contribute to every step of the way, including my postdoc advisor, Kathy Collins, Evan Nogales, and all the lab members, our collaborators at Stanford and UCSC, our colleagues at the OMB, and here are the current lab members. The people who did the work are in bold, and I already mentioned them as I go, and all our funding bodies. And thank you for your attention. I would be happy to take any questions.
Thank you so much, Kelly. That was wonderful. It's really amazing insights into how this system works. We have some questions that are coming in the chat. So I'm going to say people's names. You don't have to post your name, and they will also go into the answered section. I guess I have one quick one myself. You commented that you didn't expect to get such high resolution, right? Could you comment a bit more on why you got there? Why?
Yeah. Various things. So first is I think the quality of the sample is one thing. Second, we had to overcome quite a lot of technical things which I didn't go into details. That the sample has preferred orientation, so you need some treatment to get it. You know, you have all different views. That's just a technical thing. But one of the key things that allow us to get such high resolution now is the ability to collect lots of data. And that's important for sorting out the conformational heterogeneity, because actually 10 years ago, we couldn't have collected this much data at we speed. We probably spent months on the microscope 10 years ago. And now the speed of data collection improved, I would say 20-fold. So in the same amount of time, you can get 10 or 20 times more data. And that really was powerful in this case to do all the computational work. And the image processing pipeline also got a lot better to handle such large data. And it also got better because there's more samples of such conformational heterogeneity. So your algorithm gets better. So it goes really hand in hand. So it's evolved with the technology beside the sample preparation, yeah.
And do you see it still continuing then? Because, you know, how far can we go?
Yeah, I mean, currently we're not really using for this last one, we're not using the best of the best, but we just bought that setting here and it's being installed. So I'm very excited to try out, you know, even the latest technology that people are able to get down to 1.2 angstrom. So yeah.
Wonderful. Wow. Okay. So I have a question here from Quentin Smith. From the 8 angstrom structure, which had 1 RNA and 10 proteins, do you see dynamics, intermediate structures, without several domains, or is the telomerase complex stable once complete? I guess that was the thing I wondered about too, is sort of dynamics and intermediates of the system.
It's a good question. So the way we purify is really try to ensure that it's a complete complex before we put it on the grid. So we don't see so much, as I mentioned, it's quite compositionally homogeneous. So we eliminate part of the problem already in the purification method. So yeah.
Kelly, can I follow up on this? And also, you know, following up on Liz's previous question, the amount of data. I mean, you mentioned that in your, previous work where you resolved the structure to 8 angstrom, you only used 3 to 4% of the particles.
Yeah.
So do you understand whether, you know, the remaining part of the large majority of the particles are really, you know, just too flexible to resolve? And does that mean that now that you have much larger amounts of data, you might be able to resolve different conformations?
Yeah, that's a great question. So there's a few things. A bit is we always get some bad particles when you make EM grid because of the way we handle it's a bit harsh. But most of it is really the conformational, because we could never get the two together in one good resolution. But each of them has hinging motion. And we have the DNA in the sample, and the DNA occupancy is not always 100%. So there's a bit of heterogeneity there, and the domains also flexing around. So what we actually observe when I was using a recent analysis that CryoDragon, the Elyson, we took that subset with the heavily filter. There's still so much heterogeneity there. Okay. There's still things that are coming in and out that we couldn't assign. So there's so much more in the data. I can imagine if we have even 10 times more data, we probably can resolve more cool things that, the histones, for example. Not every particle has a histone, so we really had to kind of go to that subset that has histones, So then what else is there? We consider it incomplete, that's why I didn't focus on it, but it's yeah, there's a lot of stuff in the data that we now can go back and resolve. Yeah.
Beautiful work.
We have a related question from Eva Nogales. So relating to the histones, are they expected to be present in all telomerases across species? How are you testing the functional relevance of what they may be doing?
Yeah, so regarding across different species, telomerase is very divergent across different species. And for example, tetrahymena, where there's been very structurally well characterized, like the level of human telomerase, that same region is more minimal, so it's bound by something else, that's not histones. Yeast and the fungi family has the same three-way junction, but it's not known whether it's bound to anything there. But there's no structure of yeast, or even the complete composition of the yeast enzyme is not known. So that's something that one could try to do in the future. In terms of human, there's a few possibilities why the histone are there, which we are now trying to following up. One is that there could be a cell cycle regulation thing, because not every telomerase particle is bound. So it could be a subset of telomerase that get to the telomeres, the histones, for example. Or it could be a sub-localization of telomerase in the nucleus. Because as I mentioned, most of it is in the Cajal body. But we don't know whether the population in the Cajal body is with or without histones. So there could be various possibilities why they could come about. And we are now trying to dissect the pathway as we speak, yeah.
You can also pick some yourself, but you're busy answering. So I have one here from Jane Richardson. What does the single-stranded RNA look like that mimics what the histones see in the nucleosomes?
Oh, the RNA. So it seemed to bind to a curved backbone of RNA. So even though the RNA is kind of more flexible than DNA, but the two stem kind of brought together in a way that is curved. So it's not like a continuous DNA like in the nucleosome, but it's really curved. And we actually found some like base-specific interactions, and we're mutating them now, and they actually affect telomerase assembled in vivo. So there is an effect. So yeah, but most of it is backbone, so it's difficult to nail down. But there's just a couple of regions where we can make site-specific mutations. So
I guess I have a related question there that I wonder about. So you've talked about image processing. I'm wondering also about synergy with other methods, right? Computational predictions, other structural data to answer, some of these additional details, like to get at specific, higher resolution.
Yeah.
And dynamics.
Yeah, we're working very closely with Shaw Shearers, who developed Relion, who's down the corridor, about the dynamics, because sometimes if they have new algorithm, we usually volunteer to put our data through because we have so much data. And we also, it will come up with new conformation or variability analysis like CryoDragon or CryoSpark. We always just put our data through and see. And there's always something else. Every structure you saw is satisfying, but there's always some unmodeled density. And it's still the case for the recruitment complex. We have density in there we can't interpret. And now we keep collecting data, and hopefully one day we have enough to resolve that. So it's cool. There's always something new to follow. Yeah.
We won't be able to answer all the questions. I apologize that we can't, but we had a bit more time, so I think we'll ask a couple more. Vikash Jha, great talk. I wanted to know from your experience what type of grid is most suitable to obtain better signal-to-noise ratio for macromolecular complexes of 150 to 250 kilodalton size. So I guess going to another technical aspect.
That's a very good question. So we tried quite a few when I started, but for this specific sample, we use these C-flat grids, and we actually have a layer of carbon on top of the grid because the sample is so dilute. The particle doesn't make it into open holes. We had to put a layer of carbon support where the sample stick on. That reduces the contrast a bit, but it was fine. And we also, the trick to get to kind of good resolution for this is we try to image thin ice in thin ice. So as thin as we could get without destroying the sample so that we get better contrast. So there's three things: grid type, the carbon support, the hole size, because to get thin ice we also need big holes. So we use huge holes to get the ice to spread into thinner layers. So there's a few things technically we do. Yeah.
So I'll do just one last one, I think. Adriana Erica Mila. In your particles, there's DNA. Can you see longer sequences? I mean, can you see processivity in your EM sample?
Currently, we don't add any DNTPs, so we captured in a static stage with the DNA. And what we would like to do now, and hopefully we'll get some answers soon, is to actually walk through this processivity. So we having currently DNA like this, but we would like to look at it like that, and then when it is split back. So that's in the working, yeah.
Okay, so I think you can answer more of the questions in the chat as well. I think maybe we should go to Yulia now. Thank you very much, Kelly. That was so exciting. I'd love to have even longer discussions for the future, So thank you everyone for your questions. Again, I'm sorry we don't have time to answer all of them. But you can look for the answers also in the chat, which can be done also later.
Yeah, thank you.
Great, thank you, Liz. It's a pleasure to follow up on Kelly. And it is absolutely wonderful that Kelly has basically introduced the basics of CryoEM, because a lot of it will help understand what I will be talking about. So yeah, it's really a pleasure to be participating in this webinar. Thank you for organizing. Thanks to John for organizing. And I hope to sort of give you a glimpse into how my group tries to understand cellular functions, how they arise by understanding the fine conformational and compositional heterogeneity of macromolecular complexes, at the structural scale, at an atomistic scale. But what is unique is that we're trying to gain that structural understanding by looking at macromolecules directly in their native functional environment. So embedded with all neighboring macromolecular complexes in a cellular environment. And we do this simply by doing structural biology directly inside single cells or multicellular models. And so we really try to we do work on that rich interface between cell and structural biology, meaning that we need to cross this very large scale of spatial dimensions and really try to understand biology on multiple scales. And so the technique that really allows us to do this is called cryo-electron tomography, and we specifically focus on doing cryo-electron tomography in situ, inside cells. And so very much in analogy to what Kelly has shown you, we also start with a sample being vitrified and frozen in its pristine glassy state. But in our case, the sample is an intact cell. And so we can grow cells, human cells, directly on traditional EM grids. My group has been working on optimizing these methods to make them more reproducible. And so we have developed photomicropatterning where we can dictate where on a grid cells will grow. We can give them a more convenient environment where they're happy, and then apply any perturbation that we're interested in, either environmental or genetic. And then we can arrest all cellular processes at one point in time simply by plunge freezing. Now, once the cells are frozen, that structure of interest that we would like to look at is somewhere within that vast space of a eukaryotic or human cell. And so to be able to localize where structures of interest exist, we typically use fluorescence microscopy, which we do in the frozen state. And using confocal fluorescence microscopy, we're able to obtain optical sectioning and define three-dimensional coordinates where structures exist inside cells. We use this information to register our three-dimensional coordinates onto these types of images, because at the next step, we need to thin down these cells that are approximately 10 micrometers in thickness into the realm of what is approachable by transmission electron microscopy to achieve high resolution. And so the method of choice is using focused ion beam technology. And so in a frozen state, we can basically on a computer screen define patterns that allow us then to scan in a site-specific manner with a focused beam of gallium ions. These are heavy ions that when scanning through a biological sample will literally knock off single atoms from the sample. This allows us to generate these thin slices of cells that remain suspended by the remaining of the cell. And so we can create multiple of these lamella across an EM grid and then transfer the whole grid to a transmission electron microscope. Now, these preparations were quite challenging. So the first proof of concept were already suggested in 2007 that cryo-focused ion beam technology can be used to generate very good, very well preserved slices of cells, but it took quite some time, over a decade, to get these technologies to become more routine, automated, and reproducible, and now much more available to the broader life science community. Now, once these lamella are imaged in transmission electron microscope, what Kelly has shown you is that in single particle analysis, you acquire many 2D images across the whole grid and multiple locations of the grid. In tomography, we instead image the same area multiple times, but we do this while tilting the specimen, so we get different views of the same area of a cell. This allows us then to reconstruct a three-dimensional representation of how that cellular area looks like in analogy to a CT scan of a human patient. These are the raw data that we can start then processing, and this is a representation of a cellular, intracellular area in a yeast cell, for example, and it is basically a grayscale voxel representation where every single molecule that existed in that area in the cell contributed to some signal. Now, many of these molecules are too small to generate sufficient signal or to provide us enough signal to recognize their structures directly in the data, but there are many macromolecules or biological structures that are large enough to identify just based on their three-dimensional structural signatures. And so we can use that a priori knowledge. We know what biological membranes look like, we know what cytoskeletal filaments look like, or these large single blobs that are individual ribosomes. And we can use either template matching approaches to localize all these instances in our volume, or nowadays, using machine learning and training convolutional neural networks, we can actually start interpreting these data in a high throughput manner and convert these 3D grayscale voxel representations into actual annotations of biological macromolecules. This starts giving us a view of how subcellular areas are organized, but it still remained very far from the realm of what we would term structural biology. So until a few years back, we were really limited by two very critical factors when doing cryo-electron tomography. The first one is resolution, and Kelly just told you about the resolution revolution that has contributed to the emergence of single particle cryo-EM for structure determination. And cryo-electron tomography, especially the type of tomography that we do inside cell, really lagged behind. So the best we could get a few years back was maybe 12 angstrom resolution, where we can only get the envelope of a large macromolecular complex. Now, that directly contributes to a second problem is if the resolution is so low, how do we even identify what is interacting with our large macromolecular complex? And so these two fundamental limitations of cryo-electron tomography really limited the type of questions that we could address using this technology. But I was fortunate enough to have a brilliant collaborator and to engage into a project that allowed us to address these two problems and develop the methods and the frameworks that really push the limits of what you can do with cryo-electron tomography. So this project that enabled us to develop these new frameworks was done in the simplest possible self-replicating organism that exists, namely Mycoplasma of pneumonia. So Mycoplasma of pneumonia is a very small and highly genome-reduced bacterium. It only has 700 genes, and so the proteome is very much reduced in comparison to all other self-replicating organisms. And this allowed my collaborator, Yuri Rapselberg, to develop technologies that allow cross-linking, chemical cross-linking of the whole cell when it's still alive, and then going down the path to identify all possible protein-protein interactions using mass spectrometry. Now, the added value of the cross-linking is it not only tells us which two proteins interact, but it also provides some geometrical information about how the proteins interact in three-dimensional space. And so Yuri's lab, because of the highly reduced genome of Mycoplasma pneumonia, has been able to generate a whole proteome cross-linking interaction map. And we decided to focus on this subset of protein-protein interactions that I'm showing you in this diagram. And this basically shows proteins of the RNA polymerase interacting through a conserved transcription factor, NUS-A, to proteins that form the ribosome, the small ribosomal subunit. Now, to those in microbiology, this represents a long-standing question at the heart of microbiology that for decades we knew that transcription and translation, these two very fundamental processes in gene expression, should be functionally cross-talking in bacteria because these two processes are happening basically within the same compartment of a cell. They're not separated as in eukaryotes. While we knew that they functionally can crosstalk and somehow regulate each other as well, we didn't have any idea of how structurally these two complexes would interact. Now, the cross-linking mass spectrometry data strongly suggested that if they do crosstalk, they would do this through this small protein NUS-A, but actually it cannot really say that all these proteins interact at one point in time. These are binary interactions, and NUS-A could be interacting independently with the polymerase or with the ribosome. And so this is where cryo-electron tomography comes in, and I've already told you that Mycoplasma is an extremely small cell, and so this is data captured at 1.7 angstroms per pixel, showing you basically the entire cell within one field of view. The cells are 150 nanometers in thickness on average, and so they don't require thinning, allowing us to basically acquire very large-scale data. And this is exactly what Liang has done. He acquired over 350 single-cell cryotomography data sets, where in these data, I hope you can see very nicely, this is data that has already been denoised with new machine learning algorithms. You can immediately identify the individual ribosomes as extremely dense particles that you can see here, and you can even see the small and the large ribosomal subunits already in the raw data. And so what Liang did then is he went ahead and picked up all these ribosomes that occur in the data, basically summing up all 100,000 ribosomes from these single cells. We average them together. We align all these particles in three-dimensional space. We average them together to increase the signal to noise in a very similar approach to what is done in single particle analysis. But inside cells, we do expect the ribosomes and any other complex to display very large compositional and conformational heterogeneity. And so we can also go into classifications where in this case, we don't necessarily have particles that were damaged by the sample preparation or any other factors, but they will be classified based on what they're interacting with or their conformational states. And so going into this sort of hierarchical classification tree that I'm showing you here, we were very quickly fortunate enough to get maps that showed us fully assembled ribosomes that already had a very strong density associated with a small ribosomal subunit. At this point, we could focus our classifications on this density and indeed now sort out ribosomes that don't have a strong density and continue focusing more our classifications on the density associated with a small ribosomal subunit until we again reach a very small fraction from all the ribosomes that we started with in which this density was resolved to a level that at which we could very confidently fit in an RNA polymerase and could indeed show that there is a density that is unambiguously assigned to an RNA polymerase interacting with a fully assembled bacterial ribosomes. But if we actually look at now the segmented map where we fitted the RNA polymerase here in purple and the ribosome in gray, we could indeed see that there is this small green density that bridges between the two complexes and could not be explained by either of these two macromolecular machines. This is where the cross-linking data is indeed very precious, and we can use the information that we have, this three-dimensional topological information that we get from these binary cross-links to generate integrative structural models. So we can generate at the time, a homology model of this NOS8 protein that was hypothesized based on the cross-linking data, fitted together into this map, taking the crosslinks into as constraints as well, and build a fully atomic model of this new complex that we describe as an active transcription translation coupling complex. To really show that this complex is an actively transcribing and translating complex, what Liang did then, he basically applied antibiotics that stalled translation to the cells and went through exactly the same imaging and data processing analysis. And he could show that this active complex really requires active translation elongation. And if you stall translation elongation, basically the polymerases would run off while the ribosomes are stalled. On the other hand, if we stall now transcription, RNA polymerase using a small molecule, we basically have the ribosomes running into them and creating these collided complexes that have very different architectures from the actively transcribing ones. RNA polymerase is basically pushed out of that interface, and the ribosomes themselves were actually stalled because they physically get collided with the polymerases. And so this study really allowed us to basically establish a framework of how do we combine cryo-electron tomography of intact cells together with cross-linking mass spectrometry also generated from intact cells, and that captures all these transient interactions that happen inside cells to basically generate atomic models of active macromolecular complexes in their native environment. Now, what was outstanding in this study is that we had a unique opportunity in our hands. For the first time, we had a very large data set of cells acquired at quite small pixel sizes. We have picked up all these ribosomes, and especially the ones that were stalled by antibiotics, where we knew that they will be conformationally more homogeneous. And so we were able for the first time to really go and explore what is it that is limiting our resolution when we're talking about structure determination by cryo-electron tomography. And we were extremely lucky to join forces with Dmitry Teganov, who is a developer of really outstanding image processing algorithms, both for single particle cryo-EM, but also applicable to cryo-electron tomography. And Dimitri's algorithms allowed us to understand that basically our resolution in tomography was limited by the fact that while we're acquiring the same area of a cell and we're tilting the cell and we're acquiring more and more projections, the cell is being bombarded by electrons, and it would actually deform non-uniformly, non-linearly in 3 dimensions. Now, we can get a feeling for these deformations because we can see how densities of the ribosomes are moving in 3-dimensional space. We can measure them, and then we can actually correct for them, very similar to what is used in particle polishing for single particle CryoEM. And so when we revert back all these deformations and we refine our structures, we were able to generate a map of these ribosomes inside intact cells down to the physical limit of the data. So we were able to achieve 3.5 angstrom resolution for these ribosomes. We were very happy to really be able to see the small molecule antibiotics density bound to the active site in ribosome, as you would expect for this resolution. And since then, Nguyen has been able to show that even if you go to even smaller pixel sizes, you are able now to resolve maps to better than 3 angstroms if this is really what your study and your project requires. So that we have yet to hit a very strong limit of resolution in cryo-electron tomography, at least for highly abundant structures such as the ribosomes. Now, we have done this analysis for the chloramphenicol stalled ribosomes. We needed only about 20,000 ribosomes that were generated from 80 tomograms to generate this map at this resolution. We needed approximately 3 times the amount of data to generate the same resolution now for ribosomes in untreated cells, so where ribosomes are actively engaged in translation. And so using this map of now active ribosomes, Liang had to first build an atomic model because no model existed for the mycoplasma ribosome directly from this in-cell data. And this was essential because we have gone down the path of classifying now all the functional states of ribosomes based on their conformational changes. And so these are these we could classify based on the tRNA occupancy and elongation factor binding sites. And we are able now to order them into this cycle with multiple paths based on the very rich history of translation that exists both from structural biology of in vitro reconstituted complexes, but also a lot of dynamic studies that have been done in parallel by single molecule fluorescence studies. And so we were able to resolve 10 of the very major structures along translation elongation. So while the ribosome is adding a new amino acid to a forming nascent peptide. And while none of these structures is new per se, doing this directly inside cells allows us now to really classify and look at the distribution of each one of these states. So these numbers, the particle numbers that we get, do signify occupancy of each one of these states, and these do have an implication to the transition between this state and the next, or its ability to go back, and the rates of conversion from the previous state to this state. And so while we're still trying to wrap our heads around how do we transform all these different occupancies of states into an actual dynamic landscape, this data does provide also a lot of understanding of how, when you add small molecule antibiotics, how do they affect the cell beyond just binding the individual ribosome, because what we can also see is ribosomes engaged with other ribosomes have very different dynamics than ribosomes that are functioning on their own. So there's a plethora of phenomena of biological processes that we can really understand by looking at this scale from the single macromolecular complex, its local environment, but also the distribution of how this complex is functioning within each one of the cells. And we can start even thinking about single cell profiling based on functional states of individual macromolecular complexes. And so to summarize, this project with a very minimal cell model really allowed us to establish the methodology, the image processing, the data integration approaches that really allow us to bring cryo-electron tomography to a level where we can really start tackling more complex systems at higher biological complexity and address questions that we are also very much interested in, and I will be telling you about for the next few minutes. And so basically, a major focus in my group goes beyond individual macromolecular complexes, and we basically try to understand or provide an in-cell structural understanding of biomolecular condensates as a simple mechanism that is used in cell biology where cells are able to form distinct biochemical compartments without the need to basically physically segregate them by membranes or lipid bilayers from the environment. And so we study stress granules as a well-established example of cellular condensates, highly dynamic structures, and these are exemplified here in HeLa cells that are responding to heat shock. So stress granules, visualized here by a marker protein, G3BP, form upon a very fast response to stress, where G3BP basically dynamically demixes from a homogeneous solution in the cytoplasm, form these extremely dynamic spherical structures that are able to fuse, coalesce, and relax into spherical shape, bringing up this concept of liquid-like compartments in cell biology. And over the last decade, we have also contributed to understanding many of these phenomena using in vitro reconstitutions with our collaborators. And indeed, many of the proteins that we know are engaged in biomolecular condensates in vivo can form these liquid-like materials or droplets in vitro, where again these droplets can come together, fuse, and relax into spherical shape. Where if you use FRAP analysis, for example, to probe molecular exchange and dynamic, these molecules inside these droplets can diffuse and exchange very fast with their environment. But many of these proteins also have the propensity under certain conditions to transition into these amyloid-like structures that are no longer dynamics and are defined more as solid-like assemblies. Now, these kinds of observations from in vitro reconstitutions with very minimal components, maybe one or two proteins, maybe sometimes with an RNA molecule, really raise the question about what are the properties and the molecular architectures that give rise to these dynamic properties when we look at condensates in vivo. And one thing to keep in mind is that each one of these compartments in living cell can segregate specifically over 100 different proteins, where many of these proteins have disordered domain that can contribute to these liquid-like large-scale assemblies, but also many of these proteins are highly structured globular proteins that we know have enzymatic activities and so on. And so to try to understand how these complex mesoscale assemblies actually look like and how do they become functional, we use the approaches that I showed you right at the beginning of correlative light and electron microscopy and focused ion beam technology to really visualize in an unbiased way the molecular organization of such condensates in the context of unperturbed cells. And so when we started visualizing areas that we identified based on fluorescence to contain stress granules, we started obtaining these types of data where I hope you can, many of you can immediately recognize textbook compartments that are membrane bound, so such as these mitochondria here that you can identify based on the double membrane and the cristae, ER membranes that are decorated by individual ribosome, and a lot of cytoskeletal filament. There are 2 microtubules here and actin filament that you can really recognize based on the structural signature. And so what we started encountering more often associated with our fluorescence signal are patches of these extremely entangled filaments. And if we just look at a snapshot of one of these filaments, you would see that they are formed out of repetitive subunit in a sort of that forms a hollow cylinder. And we can use our subtomogram averaging to now generate a three-dimensional model of these filaments, and we can indeed see that we have individual subunits that form, that basically assemble to form a helical hollow structure. And we were very intrigued by these findings because they indicated that biomolecular condensates in vivo, despite being highly dynamic, are not completely disordered, that they might actually have a structural scaffold at the core of them, but that if there is a structural scaffold, that this structure is distinctly different from what has been hypothesized to be based on amyloid-like assemblies based on beta sheet stacking. And so we were very intrigued to follow up this discovery and try to identify which out of the hundreds of human proteins that we know segregate into stress granules would give rise to these assemblies. And so we went down the path of generating cellular fractionations, and we were actually able to obtain quite pure fractions that we could basically visualize using negative staining CryoEM, and really confirm that we can obtain purified fractions of these helical assemblies. And then we subjected these fractions to proteomics analysis based on mass spectrometry. And to our big surprise, for a very long time, we could not identify a single human protein that would describe or would explain the density, the gradient density purification that we got for these assemblies. And so we were extremely puzzled for a very long time until we were able to generate these new image processing algorithms and apply them now also to helical structures. And it was really a turning point in this work where we were able to achieve a map at a resolution that was high enough that allowed us to identify secondary structure elements. Now, at this point, looking at this map, it was extremely similar to what has been described in the literature for viral nucleocapsids, and not necessarily a human supramolecular assembly. Now, with that hypothesis, we did indeed research our proteomics data and could confirm that we were actually looking at a mumps virus infection in our HeLa cells. So this is really where, obtaining a structure based on an in situ cryo-electron tomography map led to an identification of the components. Now, there were no structures for mumps virus nucleocapsids, so we went ahead and built a full atomic model. We were able to determine two conformation of these nucleocapsid subunits, in which the major difference was how much the viral RNA here in yellow was exposed to the environment on the nucleocapsid surface. Now, that was very intriguing, these two conformations, because this data comes from what was happening directly inside the cell. And so if we step and look back at where we generated these structures from, what we could see is that now that we knew that these are viral nucleocapsid, you could see that those nucleocapsids were actually looking very different in cells that were happy, that were unstressed, and they were changing significantly as we were applying stress to the cells, in our view, to investigate what stress granules look like. So the nucleocapsid became more and more straight, so they really changed morphology, and they also became more and more crowded in their local environment. Now, I don't have the time to tell you the whole story, but basically what we managed to show using now light microscopy is that the viral nucleocapsids don't exist just dispersed throughout the cytoplasm, that they actually concentrate inside compartments in the cells that we then termed as viral replication factories based on the literature. And indeed that these factories were changing in morphology globally at the microscopic scale as the cells were experiencing stress. We were also able to show that under this increased response to stress, the viruses were becoming more active. So this morphological change that we could see on the molecular scale, but also on the whole cell scale, was associated with an increase in both viral replication and transcription, so generating more and more viruses. Now, we have went down the path of generating proteomics data to understand what is happening on the molecular scale, what are the changes, and I would simply summarize that now in our current working model. So what we encountered completely by accident, and we have to admit this, is a situation where viruses can coexist with their human host in what is clinically termed persistent or chronic viral infection. And so in a normal culture conditions, the viruses and the host coexist. There is very basal levels of replication of the virus, and new viruses are being budded continuously. These are infectious, and they can actually generate new infection if you transfer the medium to a naive cell culture. But basically, at the host level, there is no difference. The cells continue dividing. There is no difference between cells that are infected and non-infected. But under stress to the host, we could identify a significant change in these viral replication factories that we could show are liquid-like condensates that basically segregate the viral nucleocapsids. And the viral nucleocapsids were themselves undergoing a conformational change at the molecular scale that exposes the viral RNA, the genomic RNA, to the surface. Our proteomics analysis identified a highly disordered protein of the virus called the phosphoprotein that is the major condensing or the phase separating component of the viral genome that becomes phosphorylated. And it not only serves to form this compartment, but also to, as a chaperone, to bring the viral replication machinery, the viral polymerase, to the vicinity of the viral RNA. The proteomics analysis then also showed us that there is indeed an increase in the concentration of the viral polymerase within these condensates. And we could pinpoint that this phosphorylation site using AlphaFold modeling of the chaperone, the phosphoprotein, and the viral replication machinery, we could pinpoint this phosphorylation to their interaction interface. And based on this structural modeling, we could design and generate mutants, either phosphomutants or phosphodeficient mutants, that allowed us to experimentally verify that this phosphorylation is indeed a very important component that stabilizes this complex. And eventually, it is the stabilization of this complex, increasing the concentration of the viral polymerase in these condensates, that leads to an enhanced viral replication, transcription, but also release of new viruses. And so to sum up, I think we are in a very exciting time of cryo-electron tomography, allowing us to touch on these very different levels of biological complexity, thinking about complexity from the cell model level. We're extremely happy that we can tackle questions from the simplest possible bacterium, where we look at highly conserved macromolecular machines, their functional dynamics, in an ensemble state, through single cell organisms, human cell cultures, which allow us to touch a very complex biology of host virus interactions and how these might change in response to environmental conditions. But also we're starting to touch on multicellular samples, and especially this allows us now to start looking at these types of questions in organoid models that recapitulate more faithfully human tissues and their three-dimensional organization. This complexity of biology also does translate into more complexity at the experimental level. And so it does become much more difficult when you start targeting multicellular samples using now new lift-out approaches. but these technologies are fast evolving in the field, and there are multiple new directions that show that it is promising that in the next few years we might be able to achieve these types of understandings at the molecular scale, but really connect them all the way to multicellular complexity of human samples. And so with this, I really want to thank this wonderful group of people. I will point out Liang, who spearheaded the mycoplasma work. Xiaojie has been the brave person behind the discovery of the mumps virus story. Both of them are now finishing up in my group and moving on to their independent positions. A lot of people here contributed to many of these method developments that really allow us now to do this interesting biology across scales. Wim Hagen used to be our cryo-electron microscopy wizard at EMBL, and I would thank you for your attention.
Thank you so much, Julia. That's a further building on our first talk. Is just amazing to see what kind of insights we can get in this system at a structural and functional level. So again, we have some questions here. I guess I'm going to also ask one myself. It links to some of the questions that are in the chat. So you said you were looking for amyloid, but you didn't see it. Instead, you found a viral infection, I guess.
Yes.
So what are your thoughts on, why would you or wouldn't you see amyloid, and how similar, this is a question in the chat as well, Christian Wilson has asked this, are the structures that you might see in vitro and in vivo, and when do you get crazy differences? So that's a few loaded questions there, but the amyloid aspect and in vitro versus in vivo.
Right. So, I mean, we were not specifically looking for amyloid. One of the hypotheses in the phase separation field was that these condensates in vivo, because many of them rely on highly disordered domains of proteins, which are known in neurodegeneration to contribute to the formation of amyloids. One of the hypotheses was maybe at the molecular level, this supramolecular assembly could arise from amyloid-like interaction, right? And so that's the nice thing about tomography. You don't necessarily need to know what you're looking for to start with. And you simply try to target these areas and see what is in there. Now, we didn't see amyloids because there are no amyloids in stress granules as compartments. We got distracted by the visualization of the viral particles, and it turned out to be a very interesting story on its own for biomolecular condensates, because the viral replication factories themselves are liquid-like condensates that very interestingly do harbor highly structured assembly in combination with proteins that are more disordered, that can form multivalent interactions. And to me, as a structural biologist, this is a beautiful example where there is an interplay between structure and disorder, where the structural parts are the ones that confer function to some level, whereas the disorder parts allow, the compartmentalization, the mesoscale assembly of the functional factors. Yes.
Mm-hmm. So I think, yeah, it's a tough problem. So Grigory Mishetin, I'm sorry if I'm mispronouncing, how do you determine desired Z coordinate of slicing by ion beam based on fluorescence microscopy? So technical.
Okay, so the determination of Z coordinates is based on the fact that our fluorescence imaging is done using confocal microscopy, so we have actual optical sectioning. The determination of accurate 3D coordinates is based on fiducial. So we use fluorescent beads, as it is very commonly used also at room temperature fluorescence, or even more accurately in super resolution fluorescence microscopy. You use beads to really accurately determine center coordinates. And these beads and their coordinates are then used to register between the fluorescence and the electron microscopy data. Now, once these two data sets are registered, then you can identify in the focused ion beam microscope which height is your structure in, and target that for lamella preparation. And so the full protocol and procedures are published in our eLife paper from 2021.
Okay, another one from Eva Nogales. Once you account for the nucleoprotein structures in the phase-separated areas, what remains? Also, can you get some information on the nature of those compartments that set them apart from the cytosol from their ultrastructure?
So the volume occupancy of the nucleocapsid changes as the cells and the viruses were responding to the stress. So we could see a change from quite a dilute nucleocapsid phase into something that was extremely dense nucleocapsids that were almost stacked into, a paracrystalline arrangement. And that is because there are more viral particles generated as the viruses were activated in response to stress. So we are able to quantify local volume fractions, and those were extremely high at the stress conditions. The only way that we could define a boundary between these condensates and the rest of the cytoplasm was basically simply by whether the fact that they excluded other cellular structures. So ribosomes could not enter these condensates. We had also all membrane-bound organelles sort of lining them. So they really excluded all other cellular structures, and thereby they could be distinguished based on the fact that everything else was excluded and it was only the nucleocapsids.
Okay. Thank you. A question from Robert Nyenga. What would happen to the stress granules upon addition of or treating the cells with organic solvents that dissolve such phase separations?
I think that we have done experiments using hexanediol, as is typically done for biomolecular condensates. And stress granules have been shown previously by others to dissolve using hexanediol. And in our study on the viral replication factory, we could show that they also dissolve to a similar extent as stress granules. And so the interactions that govern phase separation in this case are also true for the viral condensates.
An anonymous attendee is wondering if self-stress in general might actually induce formation of condensates, irrespective of the chronic viral infection. Like, would you see something similar for, say, the cytoskeleton?
So stress granules are a normal response of cells to stress, whether or not they're infected with viruses. It is an interesting interplay between stress and viruses. Some viruses would themselves lead to stress that would then elicit stress granules, but any cell, most eukaryotic cells are actually, would form stress granules irrespective of infection. And so what we are able to describe so far for stress granules, and I'm sorry I didn't describe that part because I thought the virus story was actually quite interesting and a lesson for life, not to assume that you know what you're looking at, is that stress granules do tend to be more amorphous density, but they do harbor highly structured complexes, translation and initiation complexes that basically get segregated in stress granules as a way to downregulate translation in stress response globally throughout the cell. So we have yet to encounter condensates that have highly, filamentous structures such as the cytoskeleton so far.
Adriana, Erica, Mila again. A curiosity, were your cells freshly purchased and grown into grids, or did they undergo several passages before transfection and visualization? In other words, when could they have been infected?
Yeah, so we managed to trace back the infection to an actual fax sorting. So these are cell lines that we were really generating to stably express our stress granule markers. And so we did the regular genetic engineering, and we needed to sort them, and we actually did manage at the end to trace back to a specific fax sorting. It was very difficult to trace it back because I mentioned, that even under happy conditions, the viruses were replicating and were generating viruses that were budding into the medium. And so these were extremely infectious. Within days of, culturing naive cells in the same environment, if you're not extremely careful, you would get a new infection in the naive cell within 4 days with no problem, right? And that's why it was a little bit difficult to trace back.
Right. Okay. Well, serendipity can be good though, right? I mean, these things you can learn from them. So it's good to keep our eyes open, as you said. Christian Wilson, why does the cytoskeleton in the cell seem to be so low populated? I would imagine a structure from the cell, you would see a lot of cytoskeleton. Thanks.
Typically, yes. And of course, if you look at, tomography data that is generated really at the nuclear periphery or at the cellular periphery, there's high abundance of actin, of microtubule, intermediate filaments. But do keep in mind that the areas that I was showing you are really areas that were mostly occupied by these other cellular compartments, the stress granules and the viral replication factories that do exclude other cellular structures.
Right. And one last question, I think. Kachi Pao. In the phase separation study, is the use of GFP-like fluorescent fusion tag causing any limitation on the target protein size? And does the tag influence phase separation? In terms of a limitation of in vitro reconstitution for phase separation, would you recommend on how to navigate the result before proceeding to in-cell study? So, yeah.
Okay, so GFP fusion tags are, we need to treat them as carefully in phase separation as in all other, biochemistry and functional assays. I think the tendency nowadays is to go more for in vitro studies, to go more for direct chemical labeling, and then really making sure that the GFP tagged protein behaves similarly to a small molecule conjugated protein in vitro. In vivo, I guess the added advantage in cryo-electron tomography is we don't necessarily need to tag the protein that, the one protein that we're interested in looking at functionally. We just need a protein marker as a probe that tells us where in our environment should we be looking at. And so all other proteins, there are other hundreds of proteins of a HeLa cells that go into that location are untagged, right? So this is where maybe tomography does have an advantage, that you see everything, not just the tagged one. In terms of the limitations of in vitro reconstitutions for phase separation, we do learn a lot from the in vitro reconstitution. It allows us to do very controlled experiments, probe individual mutations, really understand the grammar, how structured domains of a protein, their RNA binding versus the disorder domain influence either, the ability to form condensates or the dynamics and the behaviors of the condensates, their specificity. So I think that there is a lot of valuable information to understand at that level that then can inform us on what type of experiments we could do in vivo. But it needs to be taken with a grain of salt that these are always minimal reconstitutions and don't always have the full complexity of a cellular condensate.
Yeah, no, it really is a beautiful synergy in it. So,
Yes.
So I think with that, I'd like to thank you both again for fantastic talks and stimulating discussion. And I'd like to thank all our attendees as well for their questions.